off, before we get underway with stuff, I wanted to mention about the optional dissection lab. The Wednesday lab, we did reproductive system yesterday, right? Okay. So the Wednesday lab group's done with lab for the semester, and then the Monday lab group will be done with theirs this coming Monday. And the last week of lab for both of those set aside for optional dissections. And I provided you with the link through an announcement to sign up for those. And I actually had quite a number of students sign up for them over all of my classes. Usually I only have a handful, but I'm going to need to do this for on multiple days. It won't be both Monday and Wednesday. It'll be just one or the other, depending on the responses that I got. And I just have to look through, for those of you that signed up for it, you checked off which of the 4 times you would be able to do it. So I just have to look at all of the responses and figure out which days it works best for all of my students. So for those of you that signed up for it, I'll get back to you and tell you when it's going to be. What I'm going to do is to try to make each day that I run it kind of equal. I'll tell you specifically which day you're coming. If you signed up for multiple days, that doesn't, it won't work out to just say, come whichever day you want. So I have to tell the lab people how many specimens I need on a particular day. So I'm going to split up the group. It's like 15 or 16 people. So I'll split that up evenly among the days. And so I need you to come on a particular day, and I'll tell you which day that is based on the ones that you signed up for. So, you'll get something from me. Hopefully over the weekend I'll get to it. I was trying to sit down and do it this morning, but other things are going on. So, and seems like there was another thing I was going to mention, but it escapes me, so I don't know what it is. So we'll go on. We didn't quite finish off the material for the digestive system last time, so I want to say something about that before we move on to the, that's what I was going to say. I can't remember if I mentioned this on Tuesday, but I did finally finish putting all of the digestive system material into the online content folder. If you use that to sort of supplement the lecture material. I do not have the urinary system or the reproductive system stuff done. I am going to try very hard to get that stuff done. I have 3 more pictures that I have to put in for the urinary presentation, and I actually have the reproductive presentation ready to go. It's just a matter of sitting down and making recordings. But I need to just say, if you tend to rely on those things, I cannot promise that I will have those available in a timely fashion this semester. I had expected to be able to get to that stuff, but I can really only do it here on campus, and especially at the end of the semester. There's so many things going on. I just can't sit down for the time that I need to record it. So if you rely on those things, I'm afraid for the rest of the semester, I can't guarantee that I'll have them done. I'll try to, but they might not be there. So, okay. What I need to finish off with in the digestive system is actually not in the digestive system chapter, but rather I want to refer to a couple of things about metabolism and nutrition. Now, metabolism is actually sort of a bunch of different things. I kind of wish I could fit this all on the same screen, but there are catabolic reactions, which means to break things down, and anabolic reactions, which means to build things up, like making proteins out of amino acids. And all of those reactions, breaking things down and building them up, is metabolism. Just to illustrate this concept a little bit, I have two posters. I'm going to hold one of them up to make this point. This is a poster I used to have on my office wall, and when I changed offices, my wife did the interior decorating. She decided that this was not as aesthetically pleasing as she wanted to have put up on the wall. So this is one of two posters. They're both the same size, showing chemical reactions. These are all the chemical reactions that take place in the cell. Now this is actually showing the chemical reactions that take place in any cell. So it has photosynthesis and those sorts of things in here, which don't apply to human cells or animal cells in general. But this is just to give you an idea of all the different things that this is. Yeah, I gotta put it up here. It would be great if I had magnets to do this with. Stay. So this is everything. I am not going to go through all of metabolism for you. What I really want to do when I say metabolism is just talk about how we use glucose to make energy. And on this poster, that's really about 24 reactions that are just localized in this area. There's sort of a series of reactions that lead up to a cyclical reaction right here. So that's really all I'm going to talk about. And that's why I'm now on the screen here. It says carbohydrate metabolism, which is just part of this chapter. And in this section of the chapter, I'm only going to talk about sort of the beginning part. The picture that we have that starts here at the bottom of the screen is an overview of what carbohydrate metabolism I want to talk about. This is the cellular respiration reactions. Now, I'm fairly certain I did this back in the respiratory system. I introduced the idea of cellular metabolism. There's a C6H12O6 plus 6O2 yields 6 CO2 plus 6 H2O. Remember seeing that reaction? Okay, it sort of ties together all of these systems. And so this is essentially what's going on there. It's more than just one chemical reaction, like I showed on that poster. It's several, maybe a couple of dozen reactions. And we usually break it up into, well, we usually break it up into 3 parts, but this has 4, which there's a good reason for it. Out in the cytoplasm, glucose is broken down. Glucose has 6 carbons in it, so it's broken down into 2 3-carbon molecules called pyruvate. Doing that yields a little bit of ATP, and some energy can be harnessed in the form of electrons. When oxygen's present, the pyruvate moves into the mitochondria, where it gets converted. One of its carbons is given off as carbon dioxide, and then the remainder of that, which is a 2-carbon molecule, gets attached to a carrier molecule or a coenzyme, which then goes into this Krebs cycle, to be broken down and gives off the other carbons as carbon dioxide. As pyruvate is modified here, and then as it goes through the Krebs cycle, more energy comes off in the form of electrons, and in the Krebs cycle we get a little bit more ATP out of it. And then the last step is the electron transport chain. So all these electrons that are represented by the yellow arrow here feed into a series of proteins that are in the inner mitochondrial membrane, and they're used to generate ATP. usually this transformation of pyruvate is sort of stuck in with the Krebs cycle because they happen in the same place, and this is just sort of getting pyruvate ready to go into the Krebs cycle. But here they actually put it in two different boxes. And I just mentioned that because I have, there is a text slide in the presentation stuff, which I won't be pulling up, but the text slide just says glycolysis, Krebs cycle, and electron transport, but includes the transformation of pyruvate in that. So glycolysis is probably a 9 to, well, I guess it is 9 reactions. This is not representing all those reactions, but just talks sort of through the major concepts of what happens. The glucose molecule has phosphates attached to it, which requires ATP. So a phosphate's taken off of ATP, leaving an ADP, and the phosphate's attached to the glucose. And then that happens again. So there's 2 phosphates there. The glucose molecule is then broken in half, and through a series of reactions, energy is harvested in the form of electrons, and another phosphate is generated out of this and attached to this thing. So we now have 4 phosphates. The 2 original phosphates are now 1 on each of the halves of the glucose molecule. So each half picks up another phosphate. And then those pieces are used to build 4 ATP molecules by taking those phosphates off and making ATP. Which means that 2 ATP go into this, a couple of new phosphates are created in the process, and then those 4 phosphates are used to make ATP. So the net gain is 2 ATP for each glucose molecule. And at the end of this, we have 2 3-carbon molecules, which are pyruvate. The top of this picture, this picture kind of summarizes that. Top of it is the first part. The second one is getting the electrons out. They're showing this twice side by side because this is addressing if oxygen is present. If oxygen is present, then the pyruvates end up going into the mitochondria where they're converted and attached to the coenzyme, and then they go into the Krebs cycle. If oxygen's not present, we get the 4 ATP for the net 2 ATP out of glycolysis, but the pyruvate then gets converted into lactate or lactic acid because of the electrons that were harvested out. But when oxygen's present, those electrons end up going into the electron transport chain along with electrons that are harvested in these other steps. This picture shows what the Krebs cycle is, or the citric acid cycle, some sources will call it. And it demonstrates it for the sake of some sort of visual representation with the chemical formulae of all of the players involved. I do not want you to memorize any chemical formulae. I just want you to get the general concept of what's going on here. So when oxygen is present, pyruvate goes into the mitochondria, and this is a pyruvate molecule that has 3 carbons on it. One of those carbons comes off as carbon dioxide, and the other 2 carbons are in this fragment, which then gets attached to a carrier molecule called coenzyme A. And that goes into the Krebs cycle. So there's 2 carbons on this, and it combines with another molecule that has 4 carbons, so that we have 6 carbons. And that 6-carbon thing is modified slightly. And then 1 carbon comes off as CO2, and we have a 5-carbon molecule. Another carbon comes off as CO2, and we get to a 4-carbon molecule. And then there's a series of changes, and we get back here to the 4-carbon molecule that's going to join up with another 2 carbons from what's left over pyruvate. So this is just a a continuous loop that keeps going over and over. We get electron energy here and here. We generate actually a GTP, but that's essentially an ATP here. Another electron there, another electron there. We just keep going through and through and harvesting the energy out of this. Eventually, all of the carbons from glucose come off. So this is one pyruvate molecule. So we have 1, 2, 3 carbons that come off of one pyruvate molecule, and then there'd be another one that came from glucose, which would get rid of a 4th, 5th, and 6th carbon, which basically breaks down the glucose molecule completely, and it drives all of these reactions to get all this energy out of it. And we have all of those electrons that were harvested in glycolysis, in the transformation of pyruvate, and in the Krebs cycle. Those electrons are then used to power the pumping of hydrogen ions across the inner mitochondrial membrane. Now they just show this one membrane, but there are two, the inner mitochondrial membrane and the outer mitochondrial membrane. And between those two, there's space where these hydrogen ions accumulate, building up a concentration gradient of hydrogen ions. And so the hydrogen, to get out of this space, running down its concentration gradient, has to come out through this thing, which uses the physical movement of hydrogen through this big protein complex to take ADP and phosphates and put it together to make ATP. And that's how we make the ATP. Now, the electrons that are going through this series of proteins that are pumping the hydrogen ions across the membrane end up being used to reduce oxygen atoms. And oxygen atoms can then combine with the hydrogen ions to give us water. So carbon dioxide comes off between pyruvate being converted in the Krebs cycle, and water is made here. Those are the end results of that reaction. But really what we're doing is we're harvesting energy out of all of these molecules to drive this reaction here to make ATP. When we talk about the cellular respiration reaction, we don't talk about ADP and phosphates and ATP, because that reaction is just harvesting the energy out of those molecules, and the energy is used to make ATP through this electron transport chain. And the big thing in the membrane there that the hydrogen ions move back through is called ATP synthase. It's an enzyme that uses the physical movement of hydrogen ions to power the synthesis of ATP. So that's sort of a run through of the main things I want you to get out of this process. This picture just comes back to that original one that showed the 4 parts of what's happening here. The difference here is that it now gives you a count of ATPs that are produced. So for one glucose molecule, we make 2 ATPs, a net of 2 ATPs in glycolysis, and then we get a couple of ATPs out of the Krebs cycle. Each time it goes around, it makes 1 ATP. So for one glucose molecule, that's 2 cycles through the Krebs cycle, so 2 ATPs. And then all of the electrons that are harvested drive the production of 30 some odd ATPs in the electron transport chain. Now, they're also using the terms NADH and FADH2 occasionally. There are numbers associated with that. Those are the things that hold on to the harvested electrons. So it's not just free electrons floating around in your mitochondrion, but rather an electron is transferred from one molecule to another molecule. And those recipient molecules are NADH and FADH2. And so when they get down to the electron transport chain, they lose that electron so that they can power the hydrogen ion pumping. And at the end of that, we can get, they say 36 ATP here. Actually, that number should be a little bit of a range, because conditions matter. Under some circumstances, it'll make 36. Under others, it'll make like 34. Yeah.
NADH2 and then FADH2 transport what?
Electrons. So let me just take a second to say something about that. NAD, I don't remember what that stands for precisely, but has a positive charge. Okay, so if you add an electron to that, it neutralizes it. But what is an electron and a proton?
Atom.
Which atom is it?
Hydrogen.
Right. So NAD plus and an electron yields NAD and a hydrogen. Okay, that's why it's called NADH. And then FADH2 is FADH plus, and you add an electron to it, and it's an FADH2. So that H on there is just signifying the electron that's added to this thing that's already a positive charge. And so it gives off that electron to power the electron transport chain. That's what those terms mean. It's just the carrier molecule. For our purposes, just consider them equivalent to a spare electron that's going to be used. But for a single glucose, these are the numbers that we would expect to get out of it. If we use fatty acids or amino acids to get ATP, to make ATP, the numbers are different because it doesn't involve glycolysis and that sort of thing. But so the numbers we usually think of are based on a glucose molecule, and we can use other things to make ATP. It's a little different depending on what we start out with. Now, in the book, after talking about all that stuff, it goes on to start talking about other aspects of carbohydrate metabolism, like gluconeogenesis. We're not going to talk about that stuff. This is the only thing about metabolism that I wanted to cover. Again, it's just that cellular respiration reaction that sort of ties all of three of these chapters together. Gluconeogenesis is something that's relevant. It came up before. It's actually something that glucagon stimulates to the liver to do, so that the liver makes more glucose and it raises your blood glucose levels. But we're not going to look at the chemistry behind that. Just there are other things that are going on in our system involving carbohydrates. And then in the book here, there's a lipid and a protein section, just to say that there's metabolic processes for those. We're not going to look at them. I just want to concentrate on this process here. Okay. I also want to say a little bit about nutrition and diet. Since we've been talking about digestion and food and that sort of thing. Here in the book, they don't have a whole lot of stuff that I really care to cover. I just want to mention nutrition in the sense of what this picture is showing us. This is the My Plate depiction of balanced nutrition. Prior to this, which First Lady Michelle Obama was instrumental in developing this approach to demonstrating proper nutrition. Prior to her efforts to sort of update things, nutrition was usually depicted as the food pyramid. Originally, there was the bottom of the pyramid was made up of what we eat most of, which was grains and fruits and vegetables to get our main nutrients. And then building on that, we eat less of meats and dairy and less of sweets and oils, which is sort of the top of the pyramid. So the idea was just how much you eat going up the pyramid. But the My Plate approach is so that you can look at your plate and say, okay, I have this much meat on my plate and it represents that much of my overall meal. If your plate is just one big slab of steak, then you're not getting the proper balance in nutrition. What I want to talk about in nutrition that the book doesn't really get to is what we need to get in our diet to have proper nutrition. And there are essential nutrients that we need to get in our system. Just because I'm too. Oh, I don't want essential oils. Well, anyways, I'll go here. There are essential nutrients that we need to get in our diet because they are things that we cannot generate metabolically. We can't take other molecules and enzymatically change them to become what we need. There are essential amino acids. I like this depiction of what they are. So this list of amino acids represents the things that you have to get in your diet because we can't make it in our body from something else. And then this list here are the non-essential amino acids because we can make those from other things in our diet. Tryptophan, for instance, it has a rather complex ring structure to it, and we need to get that directly out of our diet because we can't build that out of parts of other molecules. The reason why essential amino acids is important is because every organism has what we call its protein load. Its genome codes for proteins that need to be made, and for you to be able to make the proper amount of all the proteins you need, you have to have the correct amino acids on hand. Now, protein load is a fairly broad concept. It applies to large groups. Animals all have the same protein load. Plants all have the same protein load, which means if you want to get the essential amino acids into your system through your diet, the easiest way to do that is to just eat animal proteins. And many people do this, just eating meat, you would get all of the essential amino acids that you need because a cow makes proteins just like we do, and so it's going to have those amino acids and it's the proteins that we eat in a steak or something like that. However, if you are not interested in eating animal products, you have to be aware of where you're getting your amino acids because a plant's protein load is different from an animal's protein load. So you just have to pay attention to what's in your diet. The easiest way to get the proper mix of the essential amino acids in your diet, if you're a vegetarian or a vegan, is to eat meals that are a combination of legumes, which includes beans and lentils and that kind of thing, and certain grains. So really the best vegetarian meal for essential amino acids is beans and rice, something like that. Now, if you are a vegetarian, you should know that, and if you didn't know that until I just told you now, you weren't a very good vegetarian. Because you really need to pay attention to your nutrition. And most vegetarians are aware of this, but if you're just like, I'm not going to eat things that have faces, and you say, well, I'm just going to get by on Cadbury eggs and Mountain Dew or something like that, you're not going to get all the nutrition you want, and then you're not a very good vegetarian. I don't know why I said Cadbury eggs, but whatever. Actually, I was about to go to gummy bears, and I went, well, gummy bears have faces, so that wouldn't work. But no, if you are avoiding animal products for ethical or health reasons, you do need to be aware of things like these things like essential nutrients. The other type of essential nutrient is an essential fatty acid. And essential fatty acids are, the main ones are omega-3 fatty acids and omega-6 fatty acids, and to some degree omega-9. So here, just searching for essential fatty acid, we get pictures that say omega-3 and omega-6 with pictures of food that contain those. Now, if you are a vegetarian and you're looking at this and it says omega-3 and you have just pictures of fish, and you're like, I don't eat fish, I'm a vegetarian. Fish are not the only source of omega-3 fatty acids. They're a very good source of them, and a lot of people that are trying to get in their diet and actually taking supplements will often take fish oil as a supplement. which are seemingly really disgusting big pills that just are filled with fish oil, which I think taste like fish. So just eat the fish. Yes.
Where do you find omega-9s?
Sorry?
Where do you get omega-9s in your diet?
That I know, and they're rather minor if they're considered essential fatty acids. So, but I think this picture shows pretty well. So we have a slab of fish here on the end, and then in the sort of porcelain mortar pestle thing, those are fish oil pills that are spilling out. But down here we have non-animal product examples. These are walnuts. These are almonds. Not quite sure what that is from the picture. This is supposed to be flaxseed. Oh, these are probably, no. I'm not quite sure what that is. And this is olive oil. So those are good sources of those, and probably contains omega-9s in there somewhere too. But so let me explain what omega whatever means. So first off, this picture shows us a saturated fatty acid, which is not an essential fatty acid, but I'm sort of setting up what this is. So we have a chain of carbons here with hydrogens attached to them. All of the connections from carbon to carbon are single bonds. And because of that, each of these carbons has attached to it the maximum number of hydrogens. The word saturated is referring to the fact that it's saturated with hydrogen. But really, saturated means that there are all single bonds between the carbons. An unsaturated fatty acid is one that has a double bond in it. And because of this double bond here, these two carbons only have one hydrogen attached to them, and the hydrogens that would be in this position are not. So this is unsaturated. It could have two more hydrogens in it. But really, it's called unsaturated because it has the double bond in it. So down here we have examples of essential fatty acids, which are unsaturated. They have double bonds in them. At one end of all of these fatty acids is a carbon double bonded to an oxygen and single bonded to an oxygen, which is single bonded to a hydrogen. That's COOH. It's called a carboxyl group. That's the alpha end of the fatty acid. And so the opposite end of the fatty acid is the omega end. If you count in from the omega end, the first, second, third bond is the one that's double bonded. That's an omega-3. If you go 1, 2, 3, 4, 5, 6, and the sixth one in is the first double bond. That's an omega-6 fatty acid. Now, you can keep counting on this. So that's number 3, 4, 5, 6, 7, 8, 9, and here 7, 8, 9. They're designated based on which is the first double bond. So yes, the 6th and the 9th double bonds here are double, but this is still an omega-3 fatty acid because the first one we get to is in the third bond. And that's bonds between carbons. So if you go, well, 1, 2, 3, 4, you're not counting right because that doesn't, we don't count the bond between the carbon and the hydrogen here in the end. It's carbon-carbon bond, carbon-carbon bond, carbon-carbon bond. That's double. So this is the omega-3, this is omega-6. The reason why these are important in your diet is because they are crucial for making proper phospholipids for your cell membranes. And what I'm scanning for here is I came across a good picture for this, which I put in the presentation. And I thought I just found it in searching for essential fatty acid. But so if you want to see, there's a representation. Oh, maybe it's in the book. No, it's not in the book. I don't remember where I came across, but it's in the presentation if you want to see it, just showing you a cell membrane made with just saturated fatty acids in the phospholipids, and then a representation of cell membrane made with phospholipids that have unsaturated fatty acids. The double bonds in the carbon chains force a kink in the chain. So they take up a little bit more space, and it forces the cell membrane to be more fluid. The components of the cell membrane can move around within the surface of the cell, and it's better if they're fluid. If it's made up of all saturated fatty acids, then it won't be quite as fluid. Now, in your diet, just saturated versus unsaturated, we usually associate with solid fats versus liquid fats at room temperature. So what kind of things are solid at room temperature?
Butter.
Butter. Okay. What kind of things are liquid at room temperature? Right, like corn oil or olive oil or something like that. Generally speaking, animal fats are solid at room temperature and plant oils are liquid at room temperature. So if you think about your cell membrane, if it was made up of all saturated fatty acids and the phospholipids, it would be more of a congealed cell membrane, which is not going to help cell membranes do what they need to do. So it's better to have unsaturated fatty acids in your diet for the cell membranes, and the omega-3s and omega-6s are crucial to make the proper phospholipids. So, I noticed, I said this earlier, omega-3 and omega-6, omega-9s here fall under the non-essential fatty acids, just to clear that up. But, and the main omega-3s are, I'm not going to even try to pronounce that, DHA, EPA, and ALA. And fish is a great source for the first two, and sea algae for those of you that don't want to eat animals. That's a plant matter that has it. And then flax seeds are usually the go-to example of where you get ALA. But walnuts, I don't know what chia seeds are, or chai seeds. Is that like chia pet type things?
No.
No? I don't know what they
are. They're kind of like the flaxseed.
Okay, yeah. So those are the sorts of places where you get it. And then for the omega-6s, a list of sources for a lot of those, okay. Mostly oils. Yeah. Seeds are going to have some oil in them. So if you take the seeds from cotton, you can get some cotton oil. But usually we think of cotton as in terms of the fibrous component we use for fabric. So those are the essential nutrients. So there's an essential nutrient that's important for proteins. There's an essential nutrient for in the lipids department. The other type of nutrient molecule is carbohydrates. There's not an essential carbohydrate in the same sense as these, but you could think of really any carbohydrate as an essential carbohydrate, because ultimately we don't make carbohydrates. We can make carbohydrates out of some other things under certain circumstances, but ultimately carbohydrates are made through photosynthesis, which obviously is done in plants. We don't talk about essential carbohydrates as a nutrient the same way we talk about essential amino acids and essential fatty acids, because everybody eats carbohydrates. Okay? Any plant matter you eat is going to have carbohydrates in it. Sometimes indigestible fiber, but you're usually going to get something in there. And if you are the sort of person that doesn't eat vegetable matter, you like your meat but not your potatoes or your green beans or whatever, there are carbohydrates in most everything you eat. And if you're that kind of person that you just skip vegetables altogether, you probably eat gummy bears and you get plenty of carbohydrates there. So so we don't really talk about essential carbohydrates, but ultimately carbohydrates are made through photosynthesis. So And we have to get that through our diet because we don't photosynthesize. But, okay, I went on a little longer than I wanted to about that stuff, but now we want to move on to the urinary system. So in lab, we considered urinalysis, and that's essentially what the first part of the book's about. The book then goes on to talk about the gross anatomy of urine transport. I don't remember if I might have said much about this in lab as I was talking about the organs of the system, but it bothers me, just because I have these sort of pet peeves, that they put this in this order. I would have started with the ureter, which is carrying the urine from the kidneys down, which then goes to the bladder and then goes to, it leaves through the urethra. The important thing to talk about here is the reflexive voiding of the bladder. It's called the micturition reflex, urination. As new books are coming out from year to year for A&P, I've seen something happen that I want to address a little bit. They kind of undersell the reflexes that are involved here. So the micturition reflex is when your bladder is filling or full, stretch receptors signal through the sacral spinal cord that you need to void your bladder. And the sacral part of the parasympathetic system sends fibers out that causes the detrusor muscle, which is the muscular wall of the bladder, to contract, and the internal urethral sphincter, which is also smooth muscle in the bladder wall, to relax so that you can void your bladder. Actually, let me go up to this picture. So the smooth muscle right at the opening here is the internal urethral sphincter, and then all of the other smooth muscle is the detrusor. If you are not in a socially acceptable place to void your bladder, you do have some voluntary control over this, in the sense that there's another sphincter. Around the urethra, the external urethral sphincter is skeletal muscle, which means that you have voluntary control over that. You do not, however, want to avoid going to the bathroom for too long, because as the bladder fills, once it's completely full, the urine will back up through the ureters, which can also expand to store urine a little bit. But you don't want it to get all the way back up to the kidneys, because that'll throw off the function of the kidneys, and without kidney function, severe problems are not far behind. So the parasympathetic system can actually overcome your voluntary control of the external urethral sphincter and cause that to relax, because when you gotta go, you gotta go. So that's what's described here. That's micturition, that's the voiding of the bladder. But there's actually another aspect to the reflex here that gets left out of these books. As the bladder's filling, there are stretch receptors in its wall. And when the bladder's full, those stretch receptors initiate the micturition reflex. But while the bladder's filling and the stretch receptors are starting to be activated, there's actually another reflex that doesn't have a formal name to it that I know of, but I call it the holding it reflex. And that's where the stretch receptors, when they're only moderately activated as the bladder's beginning to fill, they send a signal to the sacrospinal cord, but then that signal ends up going up to the sympathetic region in the thoracic and upper lumbar spinal cord. And it sends a signal down, causing the detrusor muscle to relax and the internal urethral sphincter to constrict, so that you can store urine. And once the stretch receptor gets activated enough that your bladder is getting really full, then it switches over from the sympathetic holding it to the parasympathetic voiding aspect of the reflex. And the sympathetic input is usually kind of overlooked, but this is, like many systems in your body, a place where there's competing input. So the internal urethral sphincter constricts under sympathetic control and relaxes under parasympathetic control. The detrusor muscle, which is the rest of the wall of the diaphragm, it relaxes under sympathetic control and contracts under parasympathetic control. And which of those is happening is really just a matter of how much the stretch receptors in the bladder wall are stretching. If you don't have a lot of urine in your bladder, then there's going to be a very weak signal from those stretch receptors, which leads to the sympathetic control. But once your bladder is getting very full, then the stronger signal from the stretch receptors will end up causing the parasympathetic system to do its job. And when the stretch receptors are maximally stretched, when your bladder is as full as it can get, then the parasympathetic system will actually overcome your voluntary control of the external urethral sphincter. So that's the gross anatomy of urine transport and the reflexive control of storing and eliminating urine, which basically covers everything in the urinary system except for the kidneys. Okay, so they put this up front, I guess, to kind of get it out of the way, and sort of comes right after talking about the physical characteristics of urine. But we want to now spend the rest of the time thinking about what the kidneys are and how they do their job. So I introduced what the kidneys were in lab, and I'll touch on a little bit of that, but I want to expand on things. The kidneys are located in the upper abdominal quadrants. They're posterior, they're basically in the posterior wall. They're protected by the floating ribs, it's 11th and 12th ribs, don't have cartilage that wraps around and connects to the sternum. They're just floating in back, and they act as physical barriers for the kidneys because the tubule structure in the kidneys is very fragile. And so any blunt force trauma to the kidneys can potentially cause damage. And so you want those ribs there to help protect the kidneys. There's not a picture in the book, so let me see if I can quickly pull something up. Oh, duh, I don't need to. I made it here. So what I was doing this morning was putting together the correct pictures for this presentation. So let me go in here because I have the picture I want to show you. This is a historical picture drawing from a dissection. But it shows the kidney in place within the abdominal cavity, and it's surrounded by a few important layers of tissue that help to protect it. And I put the 3 layers that I want you to be aware of down here in the bullet list. Actually, the first one isn't labeled on the picture I have here, but the fibrous capsule, right? Yeah, the fibrous capsule is connective tissue that's right around the outside of the kidney, sort of anchors it in place, fibrous connective tissue. And then it anchors it to an adipose capsule that surrounds the whole kidney. Some places will refer to that as the perinephric fat or something like that. It's just fat tissue surrounding the kidney. Usually fat tissue we think of as being a place where we store fat molecules for when we need energy in the future. And exercise is often a way to try to force our body to burn off that fat, use those calories so that we have a leaner physique. This fat is not for that purpose. This fat is cushion. So just like I was talking about, the ribs help to protect the kidneys from blunt force trauma, they're wrapped in adipose tissue, kind of like bubble wrap, to provide them with a little additional protection. If you are on some sort of crazy diet and you're just trying to get rid of as much body fat as possible, when you start to tap into the perinephric fat, you're going to put your kidneys at risk of damage more. And this fat is here not as an energy reserve so much as protection for the kidneys. So if you have one of those scales at home that when you step on it, it tells you your percent body fat, and you've decided that you're going to get your percent body fat down to, I don't know, single digits or something like that, or as close to zero as possible, then you run the risk of using this fat for energy, which is going to put your kidneys at risk for greater damage than normal. The renal fascia, the other part, is fibrous connective tissue that sort of holds all of that in place. And it's essentially this dark line here in the picture that's labeled as the anterior lamella, or layer of the renal fascia. The posterior lamella is basically right along the posterior wall. The dotted line here is the parietal layer of the peritoneum. Okay. It's what's lining the cavity that contains the digestive organs. And the kidney and a few other organs which are not in this picture are posterior to the peritoneum, and they're called retroperitoneal. They're basically incorporated into the posterior body wall. And so we see just that dotted line over the top of it, and the renal fascia sort of encapsulates all of the kidney there, holding it in place. The renal fascia also extends around the renal artery and vein, of which we see hints of cross sections here. And then over on the other side, it continues over there. But, these are layers that are holding the kidney in place and helping to protect it. Unfortunately, there's not a picture of this in the book, so I just pulled this one. Oh, I forgot to put attribution for it, but it's a public domain picture, which probably came from like Grey's Anatomy, not the television show, but the actual Dr. Gray, who was an anatomist and made this book that has all of these pictures of anatomy in it. And most of them are on Wikipedia these days because they're so old, they're public domain. Did you have a question?
Renal fascia extends around what?
So it's the black line there. So it's covering the adipose tissue that's protecting the kidney, and it continues and extends around the renal artery and vein that are coming off of the descending aorta and inferior vena cava. So all of that sort of connected together. Oh, and that's the picture of the ribs. Now I talked about kidney structure in lab. I'm not going to go into it so much. I just want to point out the pictures showing you the kidney, the cortex, the medullary pyramids. In lab, it might not have been so easy to make this point, but at the tip of each of the medullary pyramids, this thing here is called a papilla. Here's the label of it, papilla. And the opening beyond that, where the urine starts to collect, is called a minor calyx. So coming off of each papilla is a minor calyx, and then where the minor calyces come together is a major calyx, and then all of the major calyces come together at the middle, where we have the renal pelvis labeled right there. Okay. And then the renal pelvis is continuous with the ureter, because that's where the urine goes. If you're not voiding your bladder and urine is backing up through the ureter, you don't want it to come all the way back in here and get into this tissue, because that'll throw off how the kidneys filter blood. There is a valve or sphincter or something like that at the top of the ureter, which I would not really have ever known about because books never show it. But when I was teaching, I taught at a high school, and all my high school students had to do a project at the end of the semester. And one of my students was the daughter of a doctor, and so she had a connection, and she was able to observe some surgeries for people that had a problem with this valve, and they would have a greater risk of urine flooding back into the kidneys. So they had, it was an interesting project she did, explaining how the surgery is done and conditions that present with it. But most books don't mention that such a thing exists, just like most books don't talk about the upper esophageal sphincter. They are there, but they're kind of minor because for most everybody they work and they do their job, and it's rare where there's a problem with it. I just happen to have a student that did a project that highlighted them. So somewhere in here there's a valve or sphincter or something that helps to keep the urine from backing up. But it can back up and it'll throw off the problems with it. It'll cause all sorts of problems with the kidney. In lab, I ran through the names of the blood vessels that go in, and here's where we see it. This gray bar kind of shows the direction of blood flow through all of this. So blood enters through the renal artery, then goes to the segmental artery, the interlobar artery, the arcuate artery. I need to change this. Here in the book, they call the next artery the interlobular artery, which is way too close to interlobar. Okay? The only difference is there's a UL in here. This I prefer to call the cortical radiate artery. And actually, if we back up to this picture again for a second. They just call them cortical blood vessels. But these vessels extending up here are radiating out into the cortex. So I prefer the other name, which is cortical radiate. And what I really need to do is go into this picture and just put that word in here as an alternate for this, so people will know that. Interlobular arteries, cortical radiate arteries give rise to the afferent arteriole, which goes into the glomerulus, which is this unusual capillary bed here. And then the efferent arteriole comes out, and we have the peritubular capillaries, which are around the tubules of the nephron. And then venules bring the blood back to the cortical radiate vein, which the book is referring to as the interlobular vein, which then feeds into the arcuate vein and then into the interlobar vein, which finally drains out in the renal vein. Okay. Notice that there is not something called a segmental vein, even though there's a segmental artery. Don't have a reason for that, but it happens to be the case. So this is just where in the book they lay out all of those blood vessels that I talked through. And then here's a look at the structure of a nephron. Surrounding the glomerulus is the glomerular capsule, or sometimes referred to as the renal capsule, where the filtrate collects. The inner surface of the glomerular capsule is essentially the podocytes, which we'll look at later on, that are wrapped around the glomerular capillaries. The filtrate goes into these tubules, which are called the proximal convoluted tubules. Proximal because they're the first thing that filtrate flows into from the glomerulus. Convoluted because they have all the bends in them, and tubules because of the tubes. And then coming off the proximal convoluted tubule is a tubule that goes down into the medullary pyramid and loops back up. which is the loop of the nephron. Then when that gets up to the top, we go into the distal convoluted tubules, which are called distal because they're far from the last thing that the filtrate goes through of the nephron. In this picture, for the sake of what they're pointing out, they actually did something a little bit inaccurate, but for this type of picture, it's the way that you have to do it. The distal convoluted tubules actually are right next to the glomerulus. Okay? The filtrate's flowing through all of this stuff, but maybe this little bend right here should be adjacent to the glomerular capsule. Because to regulate filtration, we have specialized cells that compare what's coming out of the glomerulus to what's passing through the distal convoluted tubules, and they have to physically be next to each other. And we'll see that in another picture. But the distal, proximal and distal is just talking about the order they are in going along. The nephron ends with the distal convoluted tubule, which feeds into the collecting duct, which goes down through the medullary pyramid and releases urine into the minor calyx. There are two types of nephrons, and I made this point in lab. This picture is from the Wikipedia page on nephrons. For whatever reason, the book actually doesn't make this point. It might say something in the text, but there's not a picture for it. There are juxtamedullary nephrons and cortical nephrons. Juxtamedullary nephrons have their glomerulus and glomerular capsule right by the medullary border. Juxtamedullary means next to the medulla. And then cortical nephrons have their glomerulus higher up. I should mention this picture. Bowman's capsule is the glomerular capsule, just an old-fashioned name for it. And loop of Henle is the loop of the nephron. So cortical nephrons are just higher in the cortex, just anything that's not right next to the glomerular border. The loops are also different. The juxtamedullary nephron has a long loop that goes all the way down through the medullary pyramid, almost to the papilla. And the cortical nephrons have shorter loops that just go down a little bit and come right back up. So this picture, I think, makes a good point of those. Another thing to mention, which is not represented in this picture, is the peritubular capillaries. There's a specialization of the peritubular capillaries around the very long loop of Henle for juxtamedullary nephrons. It kind of looks like a rope ladder. It's called the vasa recta, which means right angle vessels or something like that. And it's just because it's such a long loop, there's capillaries that are sort of around it all the way down in a very regular pattern. The capillaries around the loop of the cortical nephron aren't at all different from the capillaries around other parts of the nephron. So just specialized capillaries around the longer loop of the juxtamedullary nephron. Whoops, I never got around to putting in the movement from this picture to this picture. In lab, I talked about sort of a 3-step process for making urine. This picture, which comes from, I think this is in the kidney page for Wikipedia, shows 4 steps, but, and I just didn't have time to modify the picture the way that I would. So the first thing that happens is filtration, which is everything that can be is filtered out of the blood at the glomerulus. And then the next step is reabsorption, which is where we reclaim things from the filtrate back into the blood, like glucose, amino acids, and certain electrolytes that we definitely want to keep. And then I use the word excretion. Here they're using secretion for the 3rd step, which is when we move things from the blood into the filtrate directly, because we know we don't want to keep those in the blood. And then I didn't mention the fourth thing here, which they're calling excretion, because that's just the urine going on for storage. So if you think about it, these three things here are what I talked about in the lab, 1, 2, and 3, and they together make up excretion. So when you filter the blood, you reabsorb the things you want to keep, and you secrete the things you definitely don't want to keep. All of that together leads to the production of urine, which is excreted from the kidneys. That's what the thing down at the bottom is saying. So I'm okay with this sort of presenting it as a 4-step process, but what I was concentrating on are the first 3 steps when I was talking about it. Filtration takes place at the glomerulus, and it takes place because the capillaries are wrapped by these specialized cells called podocytes, which have foot-like extensions, which is where pod and podocyte comes from, that end up kind of meshing together, leaving a little slit in between the pedicels, which is what you call little foot-like extensions, which we call a filtration slit. And I think the next picture shows, nope. Sorry, I thought there was another picture here, but I really swear there was a picture of this. Let me go to the book. I might not have made a copy of it. Let's see. Yeah, okay. For whatever reason, I forgot to copy this picture in, but this is actually a picture from the blood vessels chapter showing a fenestrated capillary. So the capillary that's in the glomerulus is a fenestrated capillary. So the filtration membrane, the capillary wall and the podocyte together, consists of holes in the capillary wall, the basement membrane, which is basically a collagen mesh, and the podocytes with their filtration slit. So anything that can fit through the fenestrations can pass through the collagen mesh and can go through the filtration slit, will come out of the blood. So large proteins, or I should say proteins in general, because they're large, and cells definitely aren't going to be able to go through that. Lipids probably aren't either, mostly because if they're too big, they're too big. But also, I don't think lipids, being hydrophobic, would be able to pass through the basement membrane very easily. So all that comes out are small hydrophilic molecules, water, obviously, glucose, amino acids, electrolytes, and waste products all come out. That's what happens in filtration. Okay, so I meant to put that picture in there just to talk about the filtration membrane a little bit more. Filtration is driven by blood pressure. So blood hydrostatic pressure. Hydrostatic is the type of pressure that blood pressure is, which means basically fluid pressing against its container. And that should be about 55 millimeters worth of mercury of pressure at the glomerulus. For most capillaries going into it, the pressure should be about 35 millimeters of mercury. So these capillaries have a higher pressure. And it has to compete against 2 other pressures. There's osmotic pressure. It's called blood colloid osmotic pressure because the things that don't get filtered out of the blood, the cells, proteins, probably lipids too, make the blood hypertonic compared to the filtrate. Because the filtrate is just the blood without those things in it. So it has less dissolved in it. And when you have a hypertonic and hypotonic solution side by side across the membrane, to equilibrate the 2 solutions, water will move back across the membrane from the hypotonic side to the hypertonic side. And so that's going to represent 30 millimeters of mercury of pressure of water trying to get back into the blood to keep the 2 equivalent. And then also there's capsular hydrostatic pressure. As the filtrate collects in the capsule, it's going to there's pressure exerted between the fluid of the filtrate and the wall of the capsule at about 15 millimeters of mercury. So if you combine those three terms, 55 minus 30, because it's an opposing force, and minus 15, another opposing force, we get a net outward pressure of 10 millimeters of mercury. That's a fairly narrow margin of pressure. And so the kidneys are in charge of blood pressure, mostly increasing blood pressure, so that if filtration drops in the kidneys can make blood pressure go up so that the filtration continues on. We describe that filtration in a mathematical sense, I suppose you could say, as a glomerular filtration rate. This table kind of defines how we figure out what the glomerular filtration rate is for your entire urinary system. You can follow through the math if you're interested in that. Just want to put something on the board to define that. We regulate glomerular filtration initially based on what's present here. So this is the beginning of the proximal convoluted tubule. The filtrate will go through that, then through the loop, and come back. And this is a tube that's either the end of the nephron loop or the beginning of the distal convoluted tubule. Essentially the same thing. The cells in this side of the tube, which are adjacent to the glomerulus, these are called macula densa cells, and they can sense the flow rate of what's being filtered out. And I think they also have sensors for sodium and chloride, just as examples of things that are dissolved in there. If the macula densa is sensing that what's going through here is not what's supposed to be going through here, then they'll stimulate these cells here, called juxtaglomerular cells, to release signals that can regulate what's going on in the glomerulus. I think the book does a good job of explaining it, but they release, actually, I think it's ATP they use as a signaling molecule, as well as this thing called adenosine, which is related to that. And nitric oxide can be released from here too, which can vary the diameters of the afferent and efferent vessels. If flow rate is going down, so that net filtration pressure is dropping off, if we dilate the afferent arteriole and constrict the efferent arteriole, that'll mean more blood goes into the glomerulus, but has a harder time getting out, which will raise the pressure in the glomerulus and increase the flow rate. If the flow rate is too high, then constricting the afferent arteriole will allow less blood into the glomerulus and it'll decrease flow rate. So locally, through data basically collected by the macula densa, these cells here can affect flow rate through the glomerulus and therefore how much is being filtered at any given time to keep it normal. If those local changes don't work, then the kidney will release a signal called renin, which will get into the bloodstream and initiate the beginning of changing a molecule called angiotensin 1 into angiotensin 2. The reason why the lungs are here is that the converting enzyme is mainly found in, as it's passing through the blood. Angiotensin 2 is a very potent vasoconstrictor, so it can raise systemic blood pressure, which will help get that glomerular pressure up to 55 millimeters of mercury when needed. Also, angiotensin II stimulates the adrenal cortex to release aldosterone. And aldosterone targets the nephron and causes it to retain sodium. When you retain sodium, you're also going to retain water. As water volume in your blood goes up, so does blood pressure. I should say blood volume goes up because you're retaining water, so does blood pressure. So this will help with systemic changes to keep blood pressure up and keep filtration up. So that's the end of that whole section. I wanted to get to that point because that wraps up the filtration part of things. And so we'll pick up with this on Tuesday, looking at absorption and secretion and then some regulation of things a little later on. So have a nice week.